start the recording. Okay, ready when you are.
All right, so thanks for coming to this webinar. And so my name is Chris Myers. I'm a professor at the University of Utah. I also have the distinction of being the chair of the Synthetic Biology Open Language. And so I've been in this role for, I think, approximately a year now. And so one thing that's very important and unique about this standard development is it's run by a community, and all of the people that are involved are elected to their positions that manage the development of the standard. In addition to myself, there are elected SBOL editors from a variety of locations, both internationally, both in academia and industry. I need to update some of these names. I just realized Zach Palchuk is our outgoing SBOL editor. He is from Zymergen. We've just brought in a new one, Prashant, and I always have trouble with his last name, it's very long, who works at Microsoft Research in Cambridge. We'd like to make sure that we always have some industrial representation on the board. Indeed, if any of you get interested, I'd encourage you to consider becoming an SBOL editor. So, as probably you're all aware, synthetic biology is a field that was developed by a number of people with the idea of basically adding the principles of engineering to genetic engineering. So even though genetic engineering has engineering in the title, it wasn't necessarily an engineering discipline. It was mostly just various experimental techniques to manipulate DNA. So in particular, synthetic biology, as was originally defined by Drew Endy, adds the notions of standards, abstraction, and decoupling. So having standards so you can describe parts and reuse parts, and like we can do in electronic engineering and other domains. Abstraction, so you don't always work with the low-level details, so you can have higher-level descriptions of systems, use modularity and hierarchy and such. And the decoupling, which I think one of the most exciting ones, which is getting to the point where people who are designing the systems don't necessarily have to be intimately familiar with the experimental processes to build the systems. The separation of concerns, I think, is very important and powerful going forward. So standards, though, are a foundational principle, as I've just said, of synthetic biology, and that's where synthetic biology open language comes in. So the synthetic biology open language was sort of originally began and discussed about 2008. This effort was initially led by a colleague of mine, Herbert Sarro, from the University of Washington, where he got together a variety of interested people that wanted to share information about their genetic designs. In particular, they were noting the fact that many of the synthetic biology papers out there that were being published were producing results that were not reproducible. People would really struggle to reproduce them in the lab simply because a lot of the information wasn't being captured and shared with the publications. So in 2011, the first version of the synthetic biology open language was released, was published a couple of years later in this article in Nature Biotechnology. One thing that I think leads to a lot of confusion, I think is a little bit of an unfortunate naming idea in our mind, is that SBOL, or the synthetic biology open language, is actually two standards. So there's the SBOL data standard, which is used to describe, among other things, the DNA sequences in our designs, the other types of molecular components, how they interact with each other. And then there's the SBOL visual standard, which in itself is a whole different separate standardization effort. And the SBOL visual standard is basically agreeing upon how we draw the diagrams for our genetic circuits. So I'm sure many of you have picked up papers in this field and have found diagrams with all sorts of different ways of representing things like promoters and coding sequences and things like that, which makes it immensely confusing. Some of us come from the field, such as myself, from electrical engineering, where if I draw a picture of an AND gate and OR gate, anybody who has any knowledge of logic design immediately knows those are AND gates and OR gates. It's a standard set of symbols that people use in their diagrams. So the promise of SBOL Visual is to provide that standard set of images that people can use in their diagrams, so it makes it easier for people to exchange these visual representations of the circuits they design. And in some respects, SBOL Visual has been one that's been uptaked somewhat the best among the community. There's a number of people who, when you ask them if they support SBOL, they say, oh yes, I use all the SBOL Visual or the SBOL glyphs in my pictures and my diagrams and my talks. What they sometimes forget is that there's also the data standard for representing information about the sequences and other information of their designs. So SBOL 1 was mostly focused on DNA level designs. And I'll get in more detail a bit later, but essentially the idea was to represent genetic sequences that were being designed by people in synthetic biology. But what SBOL version 2 was introduced to extend beyond that. So for example, as we know, our synthetic biology designs include more than just DNA. They also include proteins, metabolites, small molecules, and other compounds and components that must interact with each other to perform a function. So in SBOL version 2, you can represent any type of genetic or actually any type of molecular component in your designs. And furthermore, it started to branch into the functional layer because it's often important to represent things like this protein represses this promoter, or this protein is coded for by this coding sequence. So it allows you to do that via a construct called an interaction. So in addition to the SBOL data version 2, there was also an SBOL visual version 2 that came out in 2017. And in SBOL visual version 2, we introduced a whole slew of new DNA level glyphs. And so basically the process for this is that people in the community would propose ideas of glyphs, and there'd be debates and discussions, and then eventually we'd have votes on, yes, that is the appropriate glyph for a DNA location, a stability element, etc. And in addition to this, because SBOL data version 2 incorporates other types of molecules, we also introduced glyphs to represent things like complexes, small molecules, proteins, RNAs, etc. And then finally, we started to introduce ways of representing the interactions between these components, like inhibition with an arrow with a flat head, or stimulation, or other types of degradation or other processes. One of the things that this community is quite proud of is in 2016, ACS Synthetic Biology, one of the premier journals in this field, recommended the use of SBOL data and SBOL visual by their authors for representing information about their genetic designs. And so in particular, what they recommend is that you have your author here that's working on their article, and hopefully they use some sort of genetic design automation tools to design the circuit they're describing in their article. And then they can output that information from their tools in some language. We'd like it to be SBOL, but we recognize that many of the software tools and processes people use today are still using other standards like GenBank, FASTA, GFF, etc. So we've built converters that allow you to convert from GenBank, FASTA, and GFF3 into the SBOL data standard. So we can take whatever form your sequence and other data is in, convert that into SBOL version 2, and upload that into some data repository for sharing information about these genetic designs. And then once you've uploaded that into a data repository, what you should do is you should then indicate where that information is stored, and you can store it initially privately in some private repository, share the equivalent of like a share link for those of you that have used Google Docs, that allow you to share with the reviewers that information before the paper is published. Then the reviewers can examine that information and then decide whether or not all of the information necessary to reproduce this work has been shared with the community. That can be part of the review process. In parallel, we might, for example, use some of the tools we've developed to automatically generate an SBOL visual image that you might include in your article. And then once the article is published, that data that was initially private can then be published publicly so that others can have access to that information. And just let me remind you, at any point, if you want to ask questions, feel free to jump in at any time. I want this to be as interactive as possible. So feel free to jump in and interrupt if you have any questions at all. So yeah, go ahead.
Well, one question I might have is, you kind of mentioned these genetic circuits, which totally makes sense for in the context of your work. A lot of the times the parts we're making is, find a bunch of orthologs for a protein, and then, maybe kind of do promoter titration and stuff. So our circuits are very simple, but there's just a lot of them.
Right.
And so a lot of times it's about, kind of dealing with this, combinatorial space of, exploration and stuff.
Mm-hmm.
Does it make sense to create kind of a SBOL design for each of those, or kind of do you guys envision
So
go ahead.
absolutely, that is an important issue. And actually, I'll get to it in a little bit later, but from the DNA level, combinatorial design, SBOL includes a methodology for describing these things more effectively, because you don't really want to have, so if you were doing this with GenBank, you'd have let's say you'd have a GenBank for every single sequence of each one of these combinatorial designs. That doesn't scale very well. So one of the things that we have in SBOL version 2 is the ability to express designs with permutations. And so we've done this mostly so far at the DNA level, but there may be things at molecular level or protein level. And so that actually be a very interesting thing to come out of this is gaps. Where are there things that maybe are not, or that are important for you to be able to represent that may not be currently covered. So yeah, definitely that's something we thought about at the DNA level that isn't effectively done with the existing data standards that are out there.
And then maybe just one more question, and maybe cover it later. Like, Amyris is a little bit secretive, which is not great, and we don't publish a lot of things.
Sure.
But a lot of times where we do interact with people, where we're like sharing designs, it's more in, like kind of communicating with vendors and stuff. Like we order thousands of constructs from Twist and stuff. How do you guys like see that like relationship working? And like, is it really just we should just send the DNA sequence, or is there some advantage to getting more kind of metadata and stuff?
Right. So I think that, first of all, I think that's one of the reasons SBOL exists, is to be able to send a lot more information than just a raw DNA sequence. And some of the people like that we work with at JGI, for example, have mentioned the fact that when they get a DNA sequence that they're being asked to build, they literally will get it like in a Word document with a bunch of markups all over the place.
Yeah.
That's not effective at all for exchange. So, I mean, depending upon what type of information you want to exchange, the thing is to try to move away from doing it in Word and Excel sheets and being able to represent this information in a more computer readable form than these techniques that are currently used today. So yes, you can exchange, say, SBOL files with the information you're describing. The other thing you might have is data repositories that are internal for storing this type of information, and it's for sharing locally within your own lab. So we've developed data repositories like the SymBioHub I'll mention a bit later. We have people who are using that completely internally with no interest in publishing or sharing. I've been talking with a company in the Bay Area called Lygos, who came out of Berkeley, and they're using SymBioHub internally now. And actually, interesting enough, they've actually started contributing some code to it, which is kind of cool. But as far as they're not probably ever going to put that up somewhere that other people have access to. They just use it within their internal flows. So although I present this from an academic standpoint, we like to share and like that and stuff like that. We know that for the commercial users of this stuff, it's going to be a very different model.
Cool, thanks.
Okay, so so one of the key aspects of this, of course, is the development of these software tools. So in order to develop software tools that support the SBOL language, to make it easier for software developers, which I think most of you are, we've developed a number of libraries so that you never even have to really think about the SBOL data standard as a file format. You think about it just as a data model, and you have classes that you work with to interact with it to create constructs and structures. All of the handling of files and even some of the communication with SymBioHub is wrapped within one of these data libraries. So one of the data libraries we developed is a Java-based library, libSBOLJ, that's supported by our group at the University of Utah, and it supports everything in the SBOL data standard up to the latest version. It's also one of the most complete libraries in terms of having full validation support. It is the thing that powers this validator converter that I mentioned up here. It has conversion support from all of these various different data standards, such as GenBank, GFF, etc. And then the one that you might be more interested in, I understand a number of you are interested in Python. There's a Python library called PYSBOL, which technically right now is a swig wrapped C++ library. So it's actually, we have a C++ library for libSBOL, for SBOL, but we also have a Python wrapper around it for those using the Python language. And in the second hour of this, Bryan Bartley from BBN will give you a demonstration, and if you like, you can follow along on a tutorial using the Python library. We also have a JavaScript library, which is very useful, especially for web development. For example, this powers a lot of what we do in the SymBioHub data repository and other web applications that we've developed over time. And it's not mentioned here, but I mentioned this fellow from Microsoft Research who's recently joined the editor's team. He's created an F# Java or SBOL library. And I think somebody mentioned that you guys maybe use F# sometimes?
Yeah, our compiler is written in F# actually.
Okay, cool. So yes, there is an F# library out there. So far, I think the only users of it are Microsoft, but I know that he'd be really keen in having other people experiment with it, and it's also publicly available. All of these are open source projects, and so they're all free and open and available.
Awesome.
So with these libraries, a number of software tools have added support or support the SBOL data and visual standard. This list was compiled, I think, a couple of years ago, so it's vastly out of date. We need to add in a bit more and do probably another round of surveys. But there's a whole variety of tools, both for doing data repositories, sequence level design, etc. So basically, with all of these tools, the main goal of SBOL is to enable us to create complete synthetic biology workflows. So we have data repositories like the SymBioHub. SBOL.me might be interesting to you. It's a data repository that was built off the KEGG database. So it basically has all the information in KEGG converted into SBOL. It was developed by one of my former PhD students. And JBay developed a data repository called ICE, which supports SBOL. There's some other data repositories. IGEM doesn't strictly support SBOL, but we've converted that. I'll talk about that a bit later to support SBOL. We've developed a number of sequence editors, such as SBOL Designer. More recently, there's a tool called SBOL Canvas, which is a web-based application that's being developed by our group. Some tools like Benchling have some SBOL support, though we're leaning on them to improve it. In many of these cases, it's really driven by customer demands, of course. So do you know what sequence editing tools, or do you use sequence editing tools from commercial vendors at all?
We use a couple. I think we use Genius does a little bit of it, and then we have this kind of old tool called Clone Manager.
Okay.
But really, most of our sequence level editing happens using this GSL compiler thing.
Okay, cool. So yeah, so I mean, I think that Genius is also, so we've talked to all of these companies, and I think they all express some level of interest, but they're all kind of small fledgling companies that, you know, have to try to survive. And so getting them pushed forward has been a bit of a process. The DoD has a contract with Benchling, and they're leaning on them for us because they really want people using SBOL.
What's DoD?
Department of Defense.
Oh, DoD. Gotcha.
DoD, sorry.
Yeah.
Yeah, so they have a fairly big synthetic biology effort, and so they are using Benchling, and they want to use some of our tools like SymBioHub, et cetera. And so they're leaning on Benchling for us a little bit. So yeah, Benchling supports an old version, SBOL version 1, but they have been slow in adopting the latest version. That being said, I've actually connected Benchling to SymBioHub to be able to fetch information in and out of Benchling using their API in the past.
Awesome.
So we also have circuit design tools, like there's the Cello tool out of MIT and Boston University, which uses SBOL for its part libraries. It uses SBOL to export its designs. The iBiosim tool is a simulator and design tool developed at the University of Utah in my group. It's very standard friendly, using SBOL, SBML for computational models, et cetera. And then once you do all of your design, you can deposit things back in these data repositories. So this is the kind of the promise and the goal of having these data standards, is we don't want to be in a situation where you have to build one tool that fits everything. You want to be able to build individual tools and then be able to communicate between the tools, and that's why we are developing SBOL.
Can I ask just a generic question on
Sure.
other people in the world? Like, how do people make these parts physically?
How do they make the parts physically?
Yeah, like, so you just use this Cello thing or iBiosim, and you can obviously put the in silico representation into one of these repositories, but I mean, are they just synthesizing it, or do?
Oh, I see what you're asking. Okay, sorry. I don't get too into the wet lab weeds for me because I'm not an experimentalist, but I think, yes, so there's a variety of things. I think most of it in the academic front is still sort of traditional. They may sequence small parts, and that they probably do still a lot of the assembly manually.
Okay.
I don't think a lot of people yet, in my experience, are doing DNA synthesis of large things. They tend to still do DNA synthesis of small parts and assemble them individually using graduate students or whoever you can get. There is some efforts of automation. I know some groups that are really into trying to use automation robotics to do the assembly. But I think that the lion's share of it is still done, at least in academia, using manual procedures. I mean, maybe Ginkgo and others have probably pushed forward a lot more of the automated processes, I imagine.
Definitely. Thanks.
So I'm sure many of you may have seen this cartoon before about the fact that we really want to be careful about creating just yet another standard to add to the pile of standards. SBOL tries not to essentially reinvent things. We're not trying to basically become the one standard that rules them all. And maybe you might have seen this one too about the dangers of industry standards. SBOL is a very open, democratically developed standard. This is confusing to some people who are familiar with industry standards, but you can't just convince me of something and it becomes part of SBOL. You have to convince the community. And so the process is, if you want to get involved, you simply send an email to the SBOL editors and say, I'm interested in being a part of this. And then you get added to our mailing lists. And we're voting on a whole bunch of things for this, the new version, SBOL version 3, that we're going to be working on very heavily next month at a meeting next month. And everybody who's a member has the same vote as everybody else. It's a very open and democratically developed data standard, and we're very keen on keeping it that way. So one other aspect about the development of SBOL is that we've been very cautious about reinventing things. So for example, I've mentioned before, there's a data standard called SBML, the Systems Biology Markup Language for developing computational models. There's other data standards for doing visualization, such as the SBGN, the Systems Biology Graphical Notation. And we don't want to basically create standards where standards that work well already exist. So in order to do that, SBOL has become a member of this organization called Combine. And Combine is a group of these data standards for systems and synthetic biology that we meet together and we interact together to try to make sure that we don't just sort of invent things when acceptable standards exist. In our group, we've developed converters and model generators from where we can take SBOL level genetic designs, farm information out of a data repository, and then build a computational model that you can then simulate your genetic circuit in a simulator represented using SBML. With SBGN, as we said, they're a graphical notation for biochemical networks. Some of the visual glyphs, like the interaction glyphs that I showed you, are inspired by things in SBGN so that we can try to be interoperable with this other visualization standard. So this is the history, but what actually is SBOL? What is the SBOL data standard trying to represent? So I'm sure many of you have either used or have heard of the FASTA data standard. The FASTA data standard is a standard, literally it's just a sequence of letters, A, G, C, T in a data file. Very simple for representing a DNA sequence. But all it really gives you is a DNA sequence. It doesn't tell you anything about where the sequence came from or what the sequence is representing. Another very commonly used data standard is GenBank. In GenBank, it's slightly better because you can have the DNA sequence, but you could also annotate locations in the sequence for example, where the promoters, the ribosome binding sites, coding sequences, and terminators reside in that sequence. Not shown here, but also in common use is another data standard called GFF. And GFF is very similar to GenBank. It's slightly better in my opinion, but it's still literally just annotating sequences. So the problem with these data standards for the use in synthetic biology is they were really developed in the DNA read era. They were developed for when we were just sequencing data, sequencing and reading off DNA sequences in nature. They're not really well suited for doing design. So that's really what was the genesis of SBOL, was coming up with a data standard that could be more forward-looking for the DNA write era. So so SBOL version 1, which I said was created about 8 or 9 years ago, incorporated sort of two major enhancements over GenBank. One of them was the ability to express designs with incomplete sequence information. So for example, as shown here, I've got a promoter and I might know the DNA sequence for the promoter, and I've got a coding sequence and I might know the coding sequence for the sequence for the coding sequence. But I know that I want a ribosome binding site after my promoter and I want a terminator after my coding sequence. But I haven't yet decided which one that I actually am going to be using. So in this way, we're able to express that I've got a promoter followed by a ribosome binding site, followed by a coding sequence, followed by a terminator. So you know the general structure of your design, and later you can go back and assign the sequence. And this gives you a little bit, I think, of what you were asking about before, of this ability to mix and match in different parts and different times. And so that feature allows you, because when you're doing design, things really might be incomplete. And so this, we think, is very useful. A second big issue is that as a designer, we always approach things in this divide and conquer mentality, and we use hierarchy to break things down from big systems to smaller systems, so we get down to parts we understand how to design. So SBOL 1 allows you to express the design of a genetic circuit hierarchically. So these dashed lines indicate separate little modules. So for example, we've got one, say, genetic inverter, another genetic inverter, which are composed together, say, to build a genetic toggle switch, for example. And so this sort of hierarchical composition was built in from the very beginning. Now, SBOL version 2 moved off the DNA line. It allowed us to be able to express things that we couldn't express before, like we wanted to be able to express the proteins that are produced from the coding sequences, as well as the small molecules that are used to induce the transcription of coding sequences, as well as how things interact with each other. Like these arrows indicate this coding sequence codes for this protein represses this promoter. So, to go into a little bit more detail about the data model itself, this red box you see on the left is SBOL version 1. In SBOL version 1, you could express genetic parts or devices, and this is using something called a component definition. The component definitions could refer to the sequence of their device. So the component definition might say this is a promoter, and you might or might not have the sequence at this point. But if you do, you could refer to the sequence, and you can also annotate the sequence to indicate where various parts exist within that DNA sequence. It allows you to express sort of this part-subpart relationship. One other class that was introduced is the collection class, which was just used to group things. So you could have, for example, my IGEM promoter collection. And so you could collect all of the component definitions for all the promoters that came from IGEM, or whatever sort of organizing principle you want to use. So SBOL version 2 added these additional classes shown here in blue. There's the module definition class, which is used to describe the composition of components. So the components were also extended in version 2 to not just be DNA components, but they could be proteins, small molecules, complexes, essentially any type of object that you want to express in your design. And modules were introduced to indicate the groupings of these components to form useful genetic circuit constructs for some useful function. Module definitions also can include some functional information represented using things called interactions. And what an interaction basically does is it says, I have a relationship between this component definition and this one. So this one component definition might be a protein and the other might be a promoter, and the relationship may be this protein represses this promoter. And you can have many-to-one relationships, and so you can have like these two components bind to form a complex. Might be another interaction. The other thing that we added was the ability to express models. And so, as I mentioned before, we try not to reinvent the wheel. We just want to be able to express that there exists a model for this circuit. And this model class basically can point out to a model that might be expressed in SBML, CellML, MATLAB, you name it. So it can point out to whatever is the model for this particular device that you've designed. SBOL version 2.1 added a couple of more minor things that to these classes, basically some attribute changes. Actually, I remember one of the changes actually came out of something that came from Amyris, from one of your former colleagues, Mike Bazell, was part of SBOL version 2.1. SBOL version 2.2 added a few new classes. In particular, it added the ability to express provenance using something called the provenance ontology. So often you want to know, where did this design come from? Or you might want to say that I built this thing and it was, it came from this design, it was derived from this design. So provenance ontology is a way of expressing sort of the history of a design. The combinatorial derivation classes is one I alluded to before. I think I'll explain it in more detail a bit later. But basically what it allows you to do is express, I have a design, let's say I have a design with a specific promoter and coding sequence, but I want to try 10 different ribosome binding sites and 20 different terminators. So it allows you to express this design variability. Implementations are used to describe physical artifacts. So for example, if you've built a design and you've got it in your freezer, it's the class you use to indicate this is physically something that exists, that there's actually some copy of this that's been made. And then the attachment class was introduced as a way of linking out to external documents. So say, for example, you have a figure or flow cytometry data or some other data that's in some other format. We don't want to convert everything and somehow have SBOL sort of be a new image format or something like that. So any sort of additional metadata, or I should say any additional data that you have for your design can be linked via this attachment class. And then the current version is SBOL version 2.3. And in SBOL version 2.3, we've added 3 new classes. One is the experiment class, which is used to describe any sort of experiment you've done in your laboratory. The experimental data class, which is used to associate metadata with a particular collection of data from a particular experiment. And then the measure class, which is used to describe some sort of measurement or important parameter. For example, maybe you've measured the binding constant of a protein to a promoter, and you would express that in the measure class.
Okay.
So are there any questions about this general structure?
So I have a question.
Yes.
So the relationship between the component definition and sequence is one way, but between the annotation and the component definition is a two-way relationship. So what is the rationale behind that? I was curious.
I'm sorry, is someone maybe a little closer to the microphone repeat his question?
Sure. So there's a two-way relationship between component definition and sequence annotation.
Mm-hmm.
And, but between the sequence and component definition, it's just one way. So basically, sequence doesn't care what component has that sequence. So what made this decision?
Okay, so first of all, I should express this is a somewhat simplified version of the UML. If you look in the actual spec, there's a bit more detail. But basically, what you're seeing here is that sequence annotation class is used to express 2 types of things. So in GenBank, for example, when you say that you have a certain thing at a certain position, this is actually more appropriately known as a sequence feature. There's a feature on the sequence somewhere here, like maybe there's the start codon. You might mark the start codon. So you can annotate sequences with these sort of in-place annotations. And in that case, there would be no reference back. This link you see coming back from sequence annotation component definition wouldn't exist in that case. So those types of features are just saying there's something interesting at this position. But there's a second use for sequence annotation, which is to represent part-subpart relationships. So the component definition, let's say, might be a full genetic inverter, right? So promoter, ribosome binding site, coding sequence, terminator. The sequence annotation says the coding sequence is located at this position in the sequence, and then it points to a component definition for the coding sequence itself. So it's not the same component definition that's being referred to by this arrow back. It's the subpart of the part. Does that make sense?
Yep, makes sense.
So it's a hierarchical, it's basically expressing the hierarchy of the design. And you see the same thing with module definition. A module definition can include modules as sort of submodules. And then those submodules themselves may be defined by module definitions. And
I had, you have one component, one basic arrow on the component definition that refers back to itself, like I thought that represents the at the top. Yes. So I thought that represents the like subparts, but
you are correct. It does represent subcomponents, but this sort of indirect loop around. So this loop around says there exist subcomponents. This other loop around indicates that where the subcomponents are located in the sequence. And it's kind of tricky for me right now because we're really in the deep midst of creating SBOL3. And one of the confusions you just expressed is being fixed in SBOL3. So in SBOL3, we're not going to need to go through a sequence annotation to indicate where something is in the sequence. We're actually moving that onto that reference to say where it is, rather than having to go through the sequence annotation class for doing that.
Oh, that's interesting.
And so we're actually going to break off this arrow that you see coming here. It's not going to exist anymore. And instead, there'll only be this one directional arrow, and sequence annotation is going to be renamed sequence feature.
That's interesting. So I guess Mike, while he was here, he led the implementation of basically a database schema that loosely follows SBOL1, I think.
Right.
And, so the way that it's structured with the subcomponents is we have a component which represents your component definition, and then we have this thing called a subcomponent link. Basically takes the box sequence annotations here, and then we have mini sequence annotations that hang off the link, which is how we attach, I guess, feature level annotations, right? And they capture the base pair positioning. So
it may be the case that your internal model is a little closer to what SBOL3 is going to end up looking like.
Oh, okay. Well, that's good.
It sounds like maybe. And I hesitate. I mean, I didn't want to. I hesitate to talk too much about SBOL3 because with standards it takes a little time.
Sure.
You have to get the community to agree. We're voting on these proposals right now. In March, we're going to meet in Cambridge, England, to try to write the spec. The libraries are going to need to be rewritten. Software tools are going to need to evolve. So you might want to kind of push out of your head that I've mentioned that it's there, because it may just confuse you a little bit. Yeah. But the main goals of SBOL3 is not to sort of add radically new things that we're going to support. In fact, it technically may not support anything more than SBOL2 does when we make the transition, but to make it more intuitive and simpler. And so there's a lot of consolidation going on. For example, component definition and module definition are going to be merged into one class. So you can express, because they really are very similar in what they express. And in addition, they're not going to be called component definition, module definition. They're going to be called component, and you'll have components and subcomponents, basically. So there's a bunch of little things like that I think the next time I give this webinar, and it will be a little bit easier with some of these things. So to explain, I think.
Nice. I had just a general question. How do people use SBOL? Obviously, to have a repository or something, you probably inevitably putting a similar data model of some sort into a database of some sort, and then you obviously might want to use the XML to kind of communicate between systems or services or parties. How do you guys think about that at all? How does that kind of work?
Well, yes, of course, all the time. It's very important to understand what the use cases are. You have to have reasons for people to want to use it. Yeah. And so I've got another research grant where we have a couple experimental domain experts, and we brought them in and had them play with SBOL Canvas and SymBioHub. And, they would pepper us with questions about, well, why would I want to use this? What do I get for it? And I think that, so one thing that I've been big on is not preaching the use of SBOL, but preaching the use of tools that support SBOL. Because I don't think users of software care. They just want their tools to work together. Sure. And so you guys, as more developers of the software, you have to care and understand what SBOL gives you. But the users of the tools have sort of a different goal, different mindset that they're using to judge things, which is basically how easy is a tool to work in their workflow, and what does it give them over conventional tools like APE or other things that they've used in the past. But so I think that the main advantage is this being able to interoperate between tools. That's why we're involved in it, because we want to develop all these different tools and we want them to work together seamlessly. And so I can give you various use cases of like why it helps us. For example, in one of our projects, we're doing this computational modeling where we want to simulate genetic circuits, and we have all of the information expressed in SBOL and a data repository. We're able to use our modeling tool to mine that information to create dynamic models to simulate the behavior of the circuits before you build them. And so that that utility is what, you know, is what's really governing our interest in use of it.
Gotcha.
I see Brian's joining, so I'm probably getting behind schedule a little bit. So, but let me go into, if I could, a little bit more of the details of this data model. So, so as I said, one of the base classes, most important class, is a component definition. Every object in SBOL has an identity, which is essentially a URI, which is a unique reference identifier. And it's usually, it looks a lot like a web address, usually HTTP : something something. And then it has, it ends often with some sort of ID. Like in this case, this is from the IGEM namespace. It's the I13504 part. This is the name of the part. It has a description. So basically every class within SBOL has these three things. You have the identity, the name, and the description. But a component definition also has a type. In this case, this type is the region of DNA. And the role is it's an engineered region. So this is using the sequence ontology, which is an ontology of all sorts of different sequence features. There's like 2,800 different sequence features that you can use to express like what is interesting about this particular portion of the sequence. This component definition is actually built out of 3 components. So a component is just an instance of another component definition. So the first component here is another component definition which represents a ribosome binding site, a ribosome entry site. The second one is the coding sequence, and the third is the terminator. And so within, say, a database, you'd have actually 4 parts expressed. You've got the individual parts, and then you have the one that's the composition of the 3. Is this clear so far?
Yep.
Okay. So then the next thing you want to be able to do is say that how these parts are ordered. And so one way of doing that is in a qualitative way, which is just saying basically how they're ordered using something called a sequence constraint. So in the sequence constraint, I'm saying that the subject, which is the ribosome binding site, precedes the object, which is the coding sequence. And then I've got another constraint that says the subject, which is the coding sequence, precedes the object, which is the terminator. So these 2 constraints indicate basically the order on the DNA line that these things appear. So far, so good?
Yeah.
So at this point, I maybe don't have sequences and I haven't really placed them specifically on the sequence. But if you want to actually place them, that's where the sequence annotation comes in. So a sequence annotation allows you to say, now I've got a DNA sequence for my part over here. I'm using the IUPAC sequence code. And the sequence annotation says that this terminator is actually located at position 746 to 875. So this location class called this is a range, which indicates the range where you can find the terminator. And the terminator itself has its own sequence class indicating what's the sequence of the terminator. Now, as I mentioned, this sequence annotation plays 2 roles in SBOL2. One is to indicate basically the locations of parts, but it also indicates the locations of features. So for example, this sequence annotation says there's an assembly scar at position 738 to 745. So this is just telling me about something on the sequence that I'd like to know about. Does that make sense? Yep. So this is the feature, and this is the part subpart.
And sorry, so that was different from the sequence constraint. Like, so the sequence annotations are exact position, and the constraints are just like a total ordering?
Correct, correct. And I mean, to first degree, that's absolutely correct. So sequence constraints allow you to express relationships. Precedes is one of the most useful ones. And sequence annotations allow you to express positions, and ranges is one of the most important positions. We also have something called cuts to indicate if you want to annotate a cut site, as well. So there are other types of annotations you can have, but the most commonly used ones are these ranges to indicate the precise location of a part within a sequence, or a feature within a sequence. And then in this case, this example is showing how you can reuse components. So for example, maybe I'm going to build 2 versions of this device using 2 different promoters. So in this first one, it is actually built out of this promoter, the J23101, which precedes this engineered region. And in the second one, it's this promoter, which precedes this region. And so this composite part, has been reused in both of these 2 different devices. Okay. All right. So that's sort of the crash course on the data model for the component definitions. Next part of the data model I wanted to mention was combinatorial derivations. And this is actually a screenshot from a tool that we've developed in our group called SBOL Designer. And in this case, I think this gets back to the question you asked me before, which is I've got this device. This is actually from the Cello paper. I've got 2 promoters followed by a ribosome binding site, a coding sequence, and terminator, but I haven't yet selected which ribosome binding site. I want it to be one of these 5. And so I can annotate this as having a choice between these 5 different variations. And then similarly, the terminator's also got a similar list of parts. So if you'd done this with GenBank and assumed that you had 5 ribosome binding sites and 5 terminators, you'd have 25 GenBank files. And instead, I've just got one SBOL combinatorial derivation, which expresses all the different permutations of my design. Is this the type of thing you were asking me about a bit earlier?
Yeah, we do some of this as well. Yeah, I guess I never thought about storing the combinatorial design. It was more, we just have scripts that generate a truckload of designs. But a representation above that is kind of nice.
Yeah, so this represents it this way. We actually, within the designer tool, we actually have procedures to enumerate them. So you can actually build a whole slew of designs automatically once you've built the template to indicate all the possible designs you want. But because you can express very large data sets, you can get to a point where it's very complex to express the millions of designs and permutations you might have.
I guess maybe a follow-up question on that. So like in a case when we would use that, we might do some experiment and then pick one of those combinations as the good one moving forward, and may not care about the rest of them in the future. Is there then is each design have its kind of its own component that we then use going forward, and the other ones just kind of come along for the ride, or how does that work?
Yeah, so what you could imagine doing with this is you could say, okay, this is the scope of the things that you tried. And so you might want to store separately the scope of the things you tried so you know what you tried, right? And then you'd build maybe a separate component definition for the ones that you built. And maybe that's the only ones you keep around separately. The ones that worked, right? You might not necessarily have as much interest in the ones that didn't work, but you might want to know that you tried them so you don't try them again.
Sure. Yeah.
Is there any annotation to say that this component definition came from this combinatorial design if you do it that way?
Yes. So actually, when you use SBOL Designer to build this, it uses this provenance ontology to link things together. So you would say each of these component definitions was derived from this combinatorial derivation, so that you know that it came out of that. And in SBOL Designer itself, its enumeration capabilities make those links for you behind the scenes, so you can kind of see how all that links together.
And sorry, just so I'm clear, so you would use the provenance thing as opposed to like the collection data model or collection object?
Yeah, I mean, it's provenance gives you a little bit more precision about expressing sort of what happened. How did this work, right? And collections are kind of they're a very lightweight way of saying these are a group of related items. And that's actually a good feed into the next slide, which is a collection. And so in a collection, in this case, this is the collection of parts used in the 2016 IGEM Interlab study. And so it's basically a collection. It has similarly the identity, name, and description, but then it has a link out through this member variables of all the members to that collection. And then when we get into the functional layer, that's where module definitions come in. And so in this case, this module definition is describing now this part is actually producing green fluorescent protein. And so now I want to express that it's not just the DNA, but also what's the protein that's being produced. And so in this case, I have a functional component, which is essentially one part. And so one of my parts is this little strand of DNA. And my other part is the protein, green fluorescent protein. And then I have an interaction that says that this is a genetic production interaction, which is describing what this arrow means. And it has two participants. One participant is the template, which is this object over here, which is telling you basically instructions for building this protein. This is using something called the systems biology ontology to express these terms. And then my second participant is the product, which is this protein that's being produced. So together, this interaction expresses the relationship. This circuit, this DNA circuit, produces this protein, GFP. And then finally, this last thing I have is I have a link out here to a model object. And the model object points to a model that's stored in SymBioHub that's described in the Systems Biology Markup Language, or SBML language. And it's an OD, or continuous time model, of my genetic circuit that models the dynamics of my circuit. And this is yet another more complicated circuit. Now I've got a module definition composed of two strands of DNA. One, which is used to produce this arabinose, which is being controlled by this arabinose small molecule, and which is activating this downstream promoter. So this module definition is composed of two modules. One module is the arabinose induction unit, and the other module is the GFP producing unit. And then this part's down here is expressing how this module is constructed from one part. This promoter basically is stitched on to this module over here. And then the next class is implementation. So now let's say we've gone and built this arabinose-induced GFP unit. And so if you built it, then we use this class called the implementation class to express that it was built. So each of these implementations are three different builds of this arabinose-induced GFP unit. This arabinose-induced GFP unit was actually transformed into E. coli and grown within the M9 media. So we have another module definition to represent that the arabinose-induced GFP unit has been put into E. coli and into the M9 media. And then the media itself is another module, and then the E. coli is another component. So basically this thing is composed of my circuit, an E. coli, and a media. And so this is describing what you want to do, and then I've built it actually this way 3 times.
And for the host organism, like E. coli, do you, in some application, or do you envision like storing the complete like chromosomes of the organism as components?
Yes, I think actually in the SD2 project, this is the STARPA project that we're involved in, Bryan and I are both involved in, we do express the entire strain genome, because it might be important. If you have that information, it's often important to have that entire genome.
It was called SD2?
Yeah, the Synergistic Data and Discovery Project.
And then I guess along those lines, right? So if you're gonna integrate your part into like a genome, right? Like as more like, we don't do a lot of plasmids here, like directly to the genome. Like, do you somehow kind of like track the changes to the genome? Like, you know, kind of have.
Yes. So so it's a bit more involved than what I have in my slides, but we have come up with a way to say basically, if you want to say that you've got something integrated in the genome, you'd have a reference to the original genome, you'd have a reference to the insert, and then you would use essentially something like the sequence. You'd essentially use a sequence annotation to explain that the composite sequence is constructed by inserting this insert into this location. And there are ways of expressing, for example, maybe when you do the insert, some bases are removed or something. You can express, essentially what subset, say, of the genome remains and what subset of the insert remains when the integration is done. So you can essentially describe the assembly process directly that way.
Gotcha.
So then the next thing I wanted to mention was this provenance ontology. And so the provenance ontology is really for describing this entire design, build, test activity. So for example, you might start with a model of something you want to build. So for example, the Cello tool uses the Verilog description. Then there's a design activity where the design activity uses the model and the design artifact is generated by this design activity. And the design activity might say it's using the Cello software version 1.7 located here. So this basically captures all of the information about the design that you want. Then the artifact you produce is a genetic circuit, which is this module you have here. And then this build activity is used to describe how the physical artifact is built. So the physical artifact is generated from a build activity, and the build activity uses the design artifact. And then the build activity has with it, for example, an agent, which might be the person who actually did the experiment, a plan, which is the protocol that you use to do the experiment. So you capture precisely what they did to build it, and who did it, and when they did it. And then we might have a test activity that might be using a particular, say, that uses a particular physical artifact, such as a particular host or circuit integrated into the genome of an E. coli cell in a particular media. And then that test activity might use a particular machine, a flow cytometer or something like that, a particular protocol. That generates some experimental data, and that experimental data could be then analyzed using a learn activity, which might be some curve fitting tool or something else that you apply. So in this way, the goal is to capture sort of every step of the design process and what was done in each step of the design process as you go around the loop. So here's an example sort of illustrating all of that. So it's getting a little bit more complicated, but basically imagine that you've taken this arabinose-induced GFP circuit and you've taken an E. coli. This activity here is a build activity, which is transforming the circuit into the E. coli. That's producing this implementation, which is the transformed E. coli. That transformed E. coli and a media would be taken together to a culture activity. That culture activity is going to result in the circuit within the media, which is this thing I showed you before, the arabinose-induced E. coli circuit in M9 media. Then you might do a flow cytometry activity using this culture. That is used to generate an experiment. That experiment would have some experimental data. That experimental data may be linking out to some FCS files. And then you might have a curve fit activity that looks at the data from this experiment. And from that, you generate a measured parameter. Maybe the fluorescence or the production rate of a particular coding sequence or something like that. And then some module in your design may refer to this measurement to say that basically this measurement of this circuit, this parameter that you've defined, was actually derived through this entire process that's been captured electronically. So basically think about it in a way like taking the method section of a paper and capturing it all digitally in this form. Does this make sense?
Yeah, much more expansive than I realized.
Thank you. So you've all probably heard about IGEM, right?
Yep.
So I don't need to go into too much detail. That's me. In black, the blue dot is my son. He was in the IGEM contest in 2017, which was quite fun. So there's this registry of parts you've probably heard of. One of the things we've done is we've converted all these parts into SBOL And I don't really need to go through all the details of this. Maybe you can look at the slides later. I want to leave Bryan some time to let you show things off. But basically, we wrote a script. We converted all the IGEM parts into component definitions and sequences and components. We extracted all the sequence annotations. We mapped their types into the sequence ontology. We mapped their annotations into annotations on SBOL. And we mapped their categories into collections. And so I only introduced this but just because I think you may have some similar activity to employ if you decide, for example, to take some of your libraries and convert them into SBOL, which is basically you need to go through each of your types of objects you have and figure out, so what's the SBOL equivalent of this if you want to express that in SBOL. And I've done this countless numbers of times now in getting libraries from different people or different places. We've done this also with the AdGene data set, for example, converted that into SBOL by writing another script. So I'm going to skip over that. Now, once you have this data set and you want to share it either internally or externally, we've developed this data repository we call SymBioHub, which is used to do that. And so you can take all these parts we've derived from the IGEM data set and you can upload those into a SymBioHub. And if you go to symbiohub.org, for example, you can find the latest IGEM data set there. So there's the IGEM data set. We have a Bacillus subtilis collection. But more importantly, SymBioHub is a software that anybody can set up and stand up in their labs. I mentioned Lygos is using it internally within their labs. We use it internally within the DARPA SD2 project. We've used it in an NSF project in MIT, Boston University. So it's one of the, I think, really useful tools this community has developed so far. So that basically gets me to this end of this sort of rapid, very rapid introduction. Hopefully you are in agreement that standards are an important enabler for data sharing and reproducibility. We're hoping to develop a full standard enabled workflows. A lot more information is available from the SBOL standard website. SBOL standard has a YouTube channel, Facebook, Twitter, and Vimeo. I don't know how active those are, but they exist for SBOL. But again, I just want to end with the point that SBOL is basically, it's not me, it's not Bryan, it's not any one of us. It's a community developed effort. There's more than 145 people that are in the SBOL developers mailing list. Hopefully some of you will consider joining. 17 countries, 42 universities, and more importantly, there's also 28 companies and government labs that are part of this development process. This is out of date, but this is some of the organizations that have been involved. We've had funding from the NSF and other places in the past. And we also want to once again thank you for being part of the SBOL Industrial Consortium and helping support the development of SBOL. So again, I apologize, it's a bit of a whirlwind. There's a lot to absorb there, a bit of the fire hose, I'm afraid. But are there any parting questions before Bryan takes you on a tour of how to use it?
I have one question
Sure.
that kind of came up, especially towards the end. There's several places where ontologies are used that you refer to. I was kind of curious, just maybe why that was used as a design choice for modeling as opposed to some other method.
Right. So the key problem we have in this domain, I think, is the fact that without using ontologies, it's very hard to interoperate. So a good case in point is things like the GenBank feature set. So in GenBank, you can put a little feature annotation on that, and there's not any place I've ever found anywhere on the web that says precisely what feature names you can use. Sorry, that's the phone in my background. And I'm sure it's nobody I want to answer. But anyway, so basically there's no list of these. There's sort of a commonly accepted list of things people use. And if you use a tool like Snapgene, when you select a feature type, you pick from this sort of commonly accepted list, but nobody actually knows what the exact list is. I've tried to find this out. Worse than that, tools like Benchling, literally it's a string you put in for the type. So maybe you like to call your things coding sequences. Somebody else might like to call them CDSs, somebody open reading frames. So there's a whole variety of names people could use. And then how do you know when you want to say, give me all the coding sequences in my library, that you're going to get all of them? So that's where ontologies come in. They enable us to basically have an agreed upon vocabulary for these agreed upon vocabulary for the terms that we want to use in this field. So I think that's why we use ontologies, is to make sure that we all agree upon the definitions of our terms.
I was wondering if just briefly you could, if you know to some extent, how are some of these other gold and silver and bronze people kind of engaging with you, and how are they using the standard?
Sure. So Bryan can answer more about BBN Raytheon, but they're one of the biggest users of the standard and drivers in the community. They're involved in the STARPA project I mentioned, and many other projects in synthetic biology, and I think they use SBOL in virtually all of them. DULIX is using SBOL Visual and SBOL in some of the tooling that they develop, for example, for basically doing genetic designs. I'm not as familiar with what Twist and IDT are doing with it. Tecellogen has supported SBOL Visual from the beginning, and I think their tools also support SBOL inside. Zymergen, as I mentioned, they've been very interested. Zach Palchek, as I said, from Zymergen, has been an editor for the past couple of years. I don't know how much they're using it internally and how much they're still investigating. Shipyard Toolchains is Mike Bissell, by the way. And that's the name of his consulting company. Yeah, I think that these companies are seeing it, especially like places like Twist, IDT, and Inscripta, as ways of really exchanging with their customers more easily the types of information they wanted to get from them.
Gotcha. Cool.
Isis can probably answer more of those questions, and she, I think, engages in the meetings that these folks have periodically.
Yeah, I got you.
All right. I think I'm going to hand the floor over to Bryan. Bryan, are you ready?
I am ready.
All right.
Are you guys ready?
Ready.
I've stopped screening, so the floor is yours, Bryan.
So hello, everybody. I'm Bryan Bartley. I'm a scientist here at Raytheon BBN Technologies. I am a member, well, our company is a member of the SBOL Industrial Consortium. I worked on SBOL as a grad student, and now I continue as a developer in my professional career as a scientist. So today I wanted to give you an introduction to the Python library. And I guess, unfortunately, the best way to start out by doing that is to give you a presentation, but I will try to keep that short so you can actually get a little bit of hands-on time. But first things first, how do I share my screen with Zoom?
Bottom left, bottom right in the middle, there's a button called share.
Yeah, the green button.
Oh, okay. There's so many teleconferencing apps and
Yeah. And I'm afraid to open my laptop because the Zoom thing, but the Python notebook that is going to be written on is what I sent out. If you want to follow along or anything.
And apologies to you, Isis, because I know you've seen this before, but need to indoctrinate your fellow colleagues.
Great.
So the Python library PySBOL is closely related to the C++ library libsbol. Probably Chris gave you some background about the different kinds of libraries available, but I'll explain the relationship between those two in a moment. The current version is 2.3.3, which I believe supports, what is the current version of SBOL, Chris? Is it 2.3? Is Chris still there?
That's correct.
Yeah, okay. So you can read and write SBOL files. It interfaces with the online validation tool and the converter tool. So you can convert SBOL back and forth from GenBank in other file formats. You can interface with SynBioHub. You can do parts-based design. You know, all of those, the kind of guiding principles of SBOL, you know, parts abstraction, parts-based design, hierarchical composition, managing design, build, test, learn workflows, support for combinatorial libraries. It's got all that fun stuff. You can install it just using the pip installer. There are wheels available for 2.7, Python 2.7, Python 3.6, and now Python 3.7. Do a pip install. I guess if you're on a Mac and you want the Python 3 version, do a pip 3 install. There's also a GitHub repo that you can run the setup script there and install an egg that way, which works some of the times, but sometimes those eggs are a little out of sync with the wheels that are on pip. Documentation I'll be talking about a little bit more. So the relationship between the Python library and the C++ library is illustrated in this slide. So the Python library is actually generated from C++ code. It's basically just wrappers on top of C++ binaries, and then it's got a little bit of extra stuff sprinkled on top. There is certain reasons that went into why we did this, which in retrospect is I'm not sure that was the best decision, but so it is. So one of the ideas is that, you know, we could implement things in C and C++, and then we could generate libraries for all kinds of scripting languages. We use this tool called SWIG to generate those wrappers for different scripting languages. In theory, you can generate JavaScript and Java, but really it's Python is the most well supported by SWIG, and really it's kind of been the only thing that there's been large demand for. So the other advantage of doing it this way is that then, you know, you would have some consistency of the API and serialization. Like you would just have basically one code base that you would need to maintain, and then you could theoretically keep all these other scripting wrappers in sync. But anyways, this tool chain, it's got some drawbacks. One, that it requires, in order to maintain the library, and these are open source libraries, you know, people have to be familiar with the SWIG tool, which is, you know, pretty cool once you learn it, but there is a learning curve there, and it's kind of arcane. And then what you get, the Python wrappers, in some ways they behave more like a C++ interface rather than a Pythonic, you know, a Pythonic package or module. So anyways, because of these drawbacks, we are working here at Raytheon on re-implementing the library natively in Python. And I think we've been promising that for months, but, you know, things always take longer than you hope they will. So one of the keys to understanding, this is kind of an entry point for really learning about SBOL and how to use the library, is the API is closely married to the specification. And I don't know if Chris covered the spec document at all. I'll just show it here briefly. So you can access this on the SBOL standard website. This is what the specification document looks like. you would never read this thing from front to back. It's more of a reference document, but it defines the data model. And the way it defines the data model is there's sort of this kind of prose description of the data model and the relationships that are represented. But really, I just want to draw your attention to the diagrams, the UML diagrams. And that's really where, if you understand the diagrams, these UML diagrams, and there's just a few kind of common features of these diagrams that you need to be familiar with, but then those map to certain patterns in the Python API. So once you kind of become familiar with those patterns, then you should be able to manipulate all manner of SBOL data structures. Pardon me, I've got a knock on the door. All right, sorry about that. So besides the specification document, which, you know, no one's gonna sit down and read the spec from cover to cover, but a good way to sort of get your feet wet is to just go to the documentation and look at some of the examples and sort of, I tried to write this sort of maybe a little better than Stack Overflow, but where you get little kind of useful tidbits and examples out of it that kind of gives you a flavor for how the library works. So this is a good resource for you. So I think Chris covered this. SBOL is based on RDF and semantic web standards. And the main point about that is every object, every SBOL data object has a unique identifier or URI. So in the Python library, there's actually 3 different types of URIs you can use. The first one is what I would describe as open world URIs. And I'm not even gonna talk about that very much because they're be dragons. The open world URIs really don't have much structure at all and can sort of be unpredictable in their syntax. It's almost like just about any string could be a valid URI, but that kind of makes and you can work with those in PySBOL, but it makes everything much more difficult. By using these more structured URIs, it allows you to automate a lot of things and make things a little more user-friendly. So the first type of URI is what's called an SBOL compliant URI, and that can sort of be decomposed into these different elements. So the first is the URI scheme, which is like HTTP, but it can also be like file. I think email too is a valid scheme, but there are different schemes for URIs. We typically use HTTP. There's the namespace, display ID, which is kind of like a local ID for the object in your namespace, and then an object version. So also in PySBOL, by default, it uses these typed URIs. And again, this interacting.
Can you just repeat the last sentence or two? You cut out.
Okay. So was I talking about SBOL?
Oh, it did it again. Okay. Let's do that. Can I do it?
Hi, are you guys there?
Hi, we're here. 1 second. We're going to try to get a more stable connection. Okay. Hopefully this helps. All right. Testing 1, 2, 3. Can you say something?
Something?
All right, perfect.
Okay. Okay. Where did I cut out? I was talking about SBOL compliant URIs. Did you miss that?
It was just for like 10 seconds.
Yeah.
Type URIs.
Type directs.
Okay. So the difference between typed URIs and compliant URIs. So these typed URIs are the bane of Chris's existence because SynBioHub does not like these typed URIs. So sometimes it causes these really, you end up with these really long URIs. SynBioHub kind of does some
A detail beyond this meeting.
yeah. Anyway, so but when you're just working in the PYSBOL API, the typed URIs kind of allow for some shortcuts. The only difference is the type of the object that is being represented. So for example, component definition, that is inserted into the URI automatically. So so I think for the examples in the Python notebook, it just uses these typed URIs. One of the first things that you should do anytime you're working in your SBOL environment is you should set the default namespace. That would be, probably in your case, something like amyris.com, or, maybe amyris.com/whatever your group is. Sysbio.org is my old lab from grad school. And if you don't set this, then it will just get set to a default, which is examples.org. The example, this is an example constructor. So this is instantiating a component definition. And when you use SBOL compliant URIs, you don't have to specify the full URI of the object you're creating. You just give it this display ID. And I'll show you here in a second that it's going to auto-generate the full URI. Constructors also have required arguments. This is also something that is closely matched with the spec document. And I will show you, I think in a couple of slides, how you can tell looking at one of those UML diagrams, what is a required argument. So in this case, the type of the component definition, by type, I mean whether it's DNA, RNA, small molecule, or protein, is one of the arguments that is required. This is just a built-in constant, which expands to the full URI for the ontology term for DNA. By the way, so I say that's required, but it's actually by default. If you did leave this off, then by default, it would just instantiate a DNA component. So this is just showing you that the BioPax DNA constant, this is just a shortcut for the full URI. You could just pass this. You could pass this as a string to the constructor as well. And sometimes you may use your own custom URIs based on your own internal ontologies, and that's valid. So this is showing, so now if I just say print CD0, that will give you the URI of the object. This is also equivalent to saying print CD0.identity. We'll see some examples of that later, but this is just a short way of, if you want to inspect an object and see what its URI is, you can just print it like this. And this is also showing that now you can see that the full SBOL typed URI was auto-constructed or auto-generated. And it inserts the display ID there. The roles property, this is what, Chris may have covered this, but this is where you would supply sequence ontology terms to say, what is this particular DNA component represent. In this case, it represents a promoter. Okay, so I showed you the spec document a minute ago. So I guess I'll just go through this component definition example and kind of just explain at a high level how you read these UML diagrams. So this this box here defines the component definition class and all of its related attributes. And so a types attribute, you saw that in some of the previous slides. That's the field that gets initiated or initialized as, say, with an ontology term that says this is a DNA component. The roles, that's also an ontology term, and that's using a sequence ontology term. Next to these fields, there's the cardinality, and this relates to what I was saying before about what are required parameters versus optional parameters. So this is saying that every component definition must have a types field specified, whereas a roles field is optional, so you don't actually have to assign that a value. And again, this is showing if I had instantiated a component definition and I left off that required field, just by default, it gets initialized to be a DNA component. So there are two other types of attributes here that you need to know about. This is the most, these are the most complicated concepts, not that complicated. Once you understand this, then everything else is gravy. So there are these two types of attributes that are represented either by the closed diamond arrow or the open diamond arrow. And an open diamond arrow is a reference to another object, but that reference is by way of a URI. So here I've set this cd0.sequences attribute, and I've initialized it to point at a sequence object. But when I dereference that and I look at the value that's actually in that attribute, what it really returns is the URI, not the actual object. So that is, so any attribute that has this open diamond arrow, what you should expect is that the value it's going to contain is a URI. And so it's also legal and valid here. I could have said cd0.sequences equals and then the URI of some object. So the other type of attribute is a closed diamond arrow, and this indicates sort of compositional relationships, such that this a sequence annotation object that is related through this attribute is compositionally related. So it's an actual data structure. And if I dereference this, the values in this attribute, what I'll actually get back is a sequence annotation object. And also the lifetime of these child objects is related to the parent's lifetime. So if the parent component definition gets garbage collected, then so does the child sequence annotation. So here, this is just showing, this is creating a child sequence annotation, and it actually returns a sequence annotation object. So those are the basic sort of patterns that you need. You just kind of need to get a basic orientation to what's going on inside the library. So any questions about that before I turn to the notebook?
No, I think that made sense.
Yeah.
Okay. Okay. So, and you don't actually need to do that. You don't need to, there's nothing special about a Jupyter notebook. You could just open up your Python interpreter and follow along just as easily. It just so happened that conveniently someone helped make this notebook for me. So, so can you see it all right? Is the font big enough?
Yep. That's even better.
Okay, so this assumes that you have an account on SynBioHub, but I don't think we're actually going to go that far today. So I'm not going to go. I've truncated this tutorial, so we probably won't do that. There is the parts.xml file, the example SBOL file that I sent along with this. You will need that. And then you will need to install, do your pip install PYSBOL. Okay.
Cool. Okay. Yes, I got the SBOL.
So then what operating system are you guys on?
So I'm using a Mac, but I'm just using our Jupyter Hub environment. So I guess Linux.
Okay. Well, hopefully it the first hurdle is just getting it to import.
Right. I got past that. It worked.
Okay. Okay, that's good. Good news. So the first thing that you do is you want to set the default namespace. Okay, I'll just say sysbio.org.
Everyone with Amyris.com.
Yep. So we want a new document that's kind of our sandbox for building designs. And then we're going to want to read in a file, this example file. Okay, that seemed to work. It didn't give me any error messages. That's good. So once you have a document, then you want to know, well, how do I inspect it? How do I look at what's inside it? So there's a couple of ways of doing that. So one thing you can do is you can just say print the doc, and it will give you a summary or an inventory of all of the objects in the document. So this has got 14 component definitions and 14 sequence objects.
Yep. Do that.
So another thing you can do is you can just iterate over the document. Whoops. And so remember, if you just print out an object, it's really going to print the URI. So that gives you the URIs of all of the objects in the document.
Cool. Okay.
So if you wanted to know more, you could look at, if you wanted to know more about these objects, you could do something like this. Looks like none of them have descriptions. These, by the way, I think, if I remember correctly, these were cello parts that came out of the cello library. They're probably a bit dated now, because I know Chris and folks in SD2 have done a lot of work on adding functional information to these parts. But this was from an early SBOL version of the cello parts. So those are all of our parts in there. Okay. So now the next thing that we're going to do is get a part from SynBioHub. So basically what I'm walking you through in this tutorial is how you would create a design out of different parts. So the interface to SynBioHub, you use a what's called a part shop object. So I'm going to say part shop. And it should say in here what the URL is that you want to use.
Is that part shop class something that you can subclass with vanilla Python, or do you have to write that with C++ and SWIG?
Okay, so that's a really interesting question. So now, one of the things which, not to toot my own horn, but one thing that I am proud of is that there is a way you can subclass these in Python. And in fact, you can subclass them and create, I don't know if Chris talked about generic top levels or annotation objects, or they're called different things, but you can create your own custom SBOL objects, and you can create your own custom objects that have no relationship to existing SBOL objects, or you can subclass existing SBOL objects, and you can serialize those and deserialize it. If you're interested in that, we could communicate offline. I could send you some examples of it. I won't go into it here. It usually requires a little bit of fiddling, but it works. And it's pretty cool when it does work.
That's awesome.
Yeah.
And maybe it's just the case that, like, you know, because we kind of have our own quasi-compliant registry. I was just curious how hard it would be to point the URL at that one. But maybe it's just we need to make the service SBOL compliant.
I'd be interested in talking with you about that more. That might be, anyways, it's an interesting problem.
Okay.
The solution might not be as straightforward as we hope, but I bet we could come up with something. So you can also, you can search you can search SynBioHub. You can do this through the web interface. All of the search features that are supported by PYSBOL, you can also just go to the web interface and do that. There's kind of 3 levels of search, which if you go to that tutorial documentation page, it goes into the other ones. For example, there's an advanced search. So for example, if you wanted to search for only promoters, right, you would do a more advanced search. This search that I'm going to do is just a plain text sort of token matching search. And all it does is it searches for this token in the and then what it's going to return is a list of records. And these records are also a type of SBOL object. So I'll print out something about those. So this usually takes a second. So I'll just give it a few seconds here. So the reality is it probably found many more of these objects than what you see here. By default, it returns 25. You can configure that so that it will return more records or all the records. I can't remember how to do it off the top of my head, but again, that's that's in the documentation. But anyway, so there's a list of parts here. Some of them have description, others don't. By the way, these parts, InterLab was a study that Jake Beal did with some of the IGEM teams, and it was basically using a standard promoter measurement kit So they were trying to do standardized measurement. And so they had this set of promoters and devices that they used for that study. So that's what we're looking at here. There is one particular, oh, I want to get the URI. To get an object, to actually fetch an object from SynBioHub, you need the URI, which, so I'm going to print that out now. I don't know if it's my library that's slow or SynBioHub that's slow or what.
Yeah, I was able to pull a single part, but we ran into some trouble when we tried to do a search. So, yeah, that sort of thing.
Did you get an HTTP error?
We got like a SSL certification error.
Yeah, okay. So I'm wondering if we should try to fix that now. So you said you're on a Linux environment.
Yeah, it's running in a Docker. It's in a, I don't know, I could try putting it in a different environment if we think that's the problem.
Yeah, so the issue on Linux is that the certificates are all located in different directories depending on which kind of Linux distribution you're using. And so there's a way to configure the certificate directory. But it depends on, you know, are you on Ubuntu? Are you on Red Hat?
That is a really good question. Oh, we're on Ubuntu. Yeah.
And it's supposed to, if you have the latest version of PYSBOL, it's supposed to auto-configure that certificate path, but who knows why it didn't do that.
Well, you can keep going, and I can circle back and figure this out. I'm sure a little Google search will get me to the right answer.
There is an issue on the PYSBOL repo that is probably closed now, but it tells you what you need to do to configure the certificate path.
Okay.
It's just a one-liner if you can find it. But okay, so moving along. So I'm going to get this part here. And I'm going to get this part just because I know, because this is a pre-baked example, that's the part I want. So one of the things that's sort of unfortunate about this search is that it returns a bunch of URIs, but it doesn't tell you exactly what those objects are, what type of objects those are. And that's a way of getting around that is you would do an advanced search where you would say, just give me component definitions or just give me module definitions. In this case, using this sort of general search, you can't tell by looking at these, what type of objects these are. But I just happen to know, because this is a pre-baked example, that this is a component definition. Pardon me, I'll make this person go away. Hi, Aaron, can I call you back in a little while? 10 minutes? Okay, thanks. Bye. Okay, I'm a popular man today. Okay, so what we got when we did the search is we just got some records, but we don't have the actual object yet. So what we're going to do now is we're going to pull that object into our document. And as I mentioned before, we need the URI of the object. And basically what this does is it imports the object into your local document. So by the way, this is another way to sort of inspect the contents of your document. So previously I was just iterating over the document, which gives you all of the objects, but this will just give me component definitions.
Okay.
So then you can see my object is now living inside my local document. Okay. Okay, so now we're going to move into how do I assemble these together? Okay, so I want to get a object reference to each of these objects. So way of doing that. Whoops. So you can use the full URI. Using the full URI will always work if you want to get an object from your document. But what I also want to show you is because we're using these structured URIs, there's sort of shorthand ways of doing this where you just use the display ID or the local ID. So it kind of makes the, you know, when you're working interactively, it makes things more concise.
Can I ask you a question?
Yes, please.
So when you imported that part into your local document, so basically the type URI became a globally compliant URI. I don't see this SBOL class anymore.
Yeah, and the URI does not get changed when you import it.
Oh.
It stays in the namespace of, you know, the original authority that, you know, has ownership of it. And this gets back to what I was saying is that PYSBOL uses the typed URIs and SynBioHub just uses regular SBOL compliant URIs.
Got it. Nice.
So, okay, so hopefully this all works. I don't think it'll give any output, but hopefully that's a good thing. Okay, so now what I'm gonna do is I'm gonna take each of those, you know, components and I'm gonna assemble that hierarchically into a new component. And I'll call that my device equals. So this will instantiate a component definition. I just want to mention in the presentation I showed you earlier, you can also instantiate objects by directly calling the constructor. So this would be legal too. But if I do that, then I have an additional step where I actually have to add that component to my document if I want to do certain things with it. Sometimes it's nice to be able to just call the constructor. In other cases, it's nice to condense these two steps, you know, into this single create call. Okay, now this is the fun part. Okay, so now I'm going to create a hierarchical assemblage. So I'm using this method called a symbol primary structure. There is another method that's simply called a symbol that will put things into an abstraction hierarchy, but it doesn't put those sequence constraints between the objects that indicate that it is a sequential arrangement of those objects. So if you remember in Chris's slides, you know, he was talking about component definitions, and they have, or components and sequence constraints between those. What this method actually does is it automates the creation of all of those sequence constraints. You can also, you can use display IDs here instead of object references. That's another way of doing it. And then another thing I want to show you here is you can specify a callback function here. And so this is a built-in callback function which will assemble these objects according to the IGEM standard assembly. And so if you have some you know, assembly protocol that, you know, maybe modifies the components somehow or inserts a sequence in between them. I'm trying to think of good examples of this. It's been a long while since I've been in the lab, but you may at Amyris have your own sort of custom, you know, assembly protocols in the lab, which you could write a callback function for, and that will help you generate the final assemblage in the final target sequence. So now that I've done that, so now I can iterate through the primary structure.
Can you hear us?
Yeah,
Oh,
I can hear you.
Do you have any idea how many different assembly protocols like exist? Are there some that are defined in either SynBioHub or is part of the spec?
This is not part of this spec. This is my own secret sauce that I put into PYSBOL. I don't know if it's useful. I put it in there because I was hoping it would be useful and cool. I do know that, like at, well, talking to Mike Bissell when he was at Amyris, that, you know, they had protocols where it was like, you know, maybe you would start with these certain parts, but, you know, sequences might get inserted or deleted as the thing got assembled.
Right.
I just wanted to add, Brian, I think it is really useful. I didn't know about this, but when we were showing off our sequence editing tools, it's really important to people these are sequence accurate, which means things like scars and other things are reflected when you describe the composite sequences.
Right. Yeah. I guess because what I was going to ask, it's like if this concatenate these guys, but then yeah, like I think probably our protocol calls for, you know, generating a bunch of primers and stuff to be able to PCR them together and stuff.
Yeah.
It sounds like, you know, if you have a callback at each assembly step, maybe that's where, you know, some of that logic to figure that those other, you know, ancillary sequences come to play.
Yeah. And I bet that your real use cases would, you know, probably this isn't gonna, this is right now in sort of the example or demo stage, right? Like, it'd be interesting to know what really is useful for you guys. And because I'm, you know, I might have to, you know, modify how the callback functions work. But it'd be really cool if you guys played around with it. Whoops. So let's see. So this will return all of the component definitions in order. So then what this is showing you is here, so here's I specified 4 objects initially to put together in a primary sequence. And these BBA G002, yada, yada, these are scar sequences that are being introduced by the standard assembly. So just automatically works like that. Okay, so now this is just an abstraction hierarchy. So these are just abstract parts. component definitions with sequence ontology terms. But now we want to generate an actual sequence, a target sequence for this thing. So then the way you do that is this. You call the compile method. The compile method takes each of those parts, looks at their sequence, and then generates the new target sequence. And also, it should create a new sequence object that also
Sorry, maybe I blanked out for a second. Does the compile do the iterating through that list with the assembly callback, or what is the compile?
It doesn't. And currently, currently the compile method does not take a callback. And again, this is where I think maybe your use case might break things, because it might be necessary that the compile method also needs to support callbacks. Depending on like what stage you want to introduce new information.
I see. Oh, so the previous one where you did the create with the callback, that just basically generated a bunch of sub component definitions and their links, and then it was smart enough to introduce the scarring, and then the compile, did that just generate the sequence that got attached to the like the main component definition?
What this does is, yeah, actually, I think what you said is correct. So what this compile method does is it generates a sequence object that's associated with my device. And in that sequence object, it sets the sequence to the compiled sequence of all the subcomponents. Does that make sense? So the compile method is basically iterating through this list, and it looks at each subcomponent's sequence, and then it compiles that into the overall target sequence.
I see.
And then and that creates a sequence object, and I can do this also. So the actual sequence object that's associated with my device, you can dereference it like this, and then it has an elements attribute. This method also just returns the sequence as a string, but there is a SBOL sequence object, first class SBOL sequence object that is also created.
I see.
So the other cool thing about this compile method is that it works recursively and hierarchically. So if you had subcomponents, that you could compile sequences that way too. And that's kind of the main, that's really kind of the thing that I wanted to show off. The last thing is, you've created this new object, so you might give it a sequence ontology term. So I'm just going to assign this a sequence ontology term for gene. One of the nice things about, so with these built-in constants, there are just a few that get used a lot for like sequence ontology. And these are just kind of built in to the library. It's supposed to be convenient, but it's kind of kludgy, I think. But one of the nice things about moving to native Python is that now there are Python modules that will allow you to automate this in a way that you could just say something like my device roles equals so you could do something like that and it would return the proper URI. And so there are some bioinformatics modules that support that kind of thing, which is one of the nice things about moving to native Python. Well, actually, I guess you could probably do this with PYSBOL too, now that I mention it.
Yeah,
But yeah.
great.
Yeah, this was not something that you could easily do with the existing C++ libraries, but makes it nice. The Java library also kind of supports this kind of lookup of URIs from the sequence ontology, where you just give it the common name, the natural language description, and it will return the proper URI, which is really the way it should work.
Gotcha.
So that's it. That is my brief tour. Looks like I lost the crowd.
Kept a lot of
We got a
it.
couple of us here.
Yeah.
Thanks for hanging tough.
Yeah. No, thanks for taking us through it. Yeah, I think it's one of those things where only a few of us are on the kind of design build side. For us, it's very interesting, but for the rest of the group, they have other problems.
Yeah. One of the things that we're moving to in the SD2 program that Chris mentioned is capturing build information and quality control information. That is something that I have been interested in for years, because when I was working in the lab, it's what I really needed. And it's been years, and we're just now getting to the point where we can like really start to support those kind of applications. So I'd be happy to work with you folks on developing some of that stuff too. But yeah, anyway, thanks very much for your time. Thanks for listening.
Yeah. Just a general question. So if we wanted to adopt more of this in our pipelines or our systems in general, would you suggest the best way is to kind of make, whatever our part repository, make that SBOL compliant so you can pull parts out, and then I'm actually not sure what the first step would be. What do you guys suggest is like embracing some of this stuff? What do you do?
That's a good question.
Are you still there?
Using and
sorry, we didn't hear the beginning of that.
Mhm.
Oh, what's going on? Can you hear us?
Can you hear us? Oh no.
And I think the key is that
Oh, well, that's
Oh no.
value is connecting two tools together.
Sound is cutting in and out. Can you do a testing 1, 2, 3? Maybe get to 10.
Testing 1, 2, 3, 4, 5, 6, 7, 8, 9, 10.
All right. Yeah.
Yeah, I'm afraid we missed your answer entirely.
Okay. Is it on my end? Maybe I can put my headset back on.
Well, right now it's working great, and I'm sure we're just wrapping up. So
what I was saying is that the real utility of SBOL is connecting things. So basically demonstrating exchange. So like we're interested in SBOL so that we can have, like you saw in his demonstration, parts in SymBioHub that we can then fetch and edit. So if you imagine if you have a repository of this type of information, if you provide a means to get it out in SBOL, and then maybe you have another tool that does something with this part information, and it's able to read the SBOL, then you're demonstrating that connectivity. So that's where I think the real value is when you're actually connecting to tools. Now, the other question would be whether if any of the existing SBOL compliant tools, like Bryan mentioned, SymBioHub or others, are useful to you and can be plugins to your workflow and your process. And in that case, then having that ability having one of your tools supporting it and using one of the community built tools would also be useful. So yeah. I guess it just depends on basically looking at, exchange pain points in your workflow and experimenting with using SBOL as the glue to connect those pain points.
Gotcha. That makes sense. Cool. All right. Well, yeah, thank you both very much. This was really interesting.
Yeah, thank you guys for your time. This has been super informative, really great, and it's recorded. So I know a lot of people are gonna be looking for this recording. So thank you very much.
Yeah. So I highlighted the cool things about PYSBOL. It has its idiosyncrasies, like I said, because it's not a native Python thing, but hopefully you got enough of a good feeling. If you run into any frustrations, post an issue on the GitHub tracker, and I'm pretty responsive to those. Happy to help.
Yeah, and I would just want to also once again invite you to join the community if you want to get involved and help influence it. This is a good time to get involved because we are moving towards the next version, which allows us to make radical changes. And also, if you have any questions that you hadn't thought of today, reach out to us at any time.
Awesome. Will do.
Great. Thanks, everyone.
Yeah, thank you again.
Yep. Have a nice one.
Thanks. Bye.